So I'm excited to be here today to share some information with you about PI3R1 and its mutations and their implications in endometrial cancers. Now, before I dive into the meat of the PI3R1 part of the talk, I wanted to just give some background on Intermountain Health, who we are, and what we do. Now is this lining up? Should we double? Yeah, I just realized that. So Intermountain Health is a large not-for-profit network of hospitals that aims to provide state-of-the-art personalized patient care. And to do this, our subdivision, Intermountain Precision Genomics, or IPG, is transforming healthcare by providing an array of genomics testing and analyses for our patients to deliver the highest quality care at some of the lowest costs in the nation, helping people live the healthiest lives possible. We provide multiple genetic services, including pharmacogenomics analysis to help tailor a patient's medications to their own genetics. We also provide genetic counseling services to our patients so they can better understand their own family medical histories, things they may pass on to their children, and they can better understand their own health as well. What my talk will focus on mainly today is this centerpiece, the precision cancer care, where myself and many others within IPG can assess a patient's tumor for different genetic mutations that may qualify them for targeted therapy or clinical trials. And we do this using our flagship precision genetics test, TerraMap, which is run on the Illumina TSO 500 platform to identify different DNA and RNA variants that may qualify a patient for the aforementioned therapies and clinical trials. Now, this testing is approved for all solid tumor types, and one of the tumor types that we see recurrently with our patients is endometrial cancer. I don't think I need to convince anyone in this room that endometrial cancer is a horrible disease, but I did want to provide some statistics. It is the most common gynecological malignancy diagnosed in Europe and the US. And in the US alone last year, there were an estimated 66,000 new cases with an associated 13,000 deaths. Now, if we translate this to Europe, they've got roughly double the population we do, and we see that ratio kind of continue where they have double the new cancer diagnoses and double deaths. And when we crunch these numbers, it comes out to about 4 to 5% of patients dying from this disease, which is really highlighting the need to understand the genetic underpinnings of this disease so we can prevent that in the future. There are already some genes implicated within endometrial cancer. For example, the POLE gene, mutations in this gene are associated with a hypermutated phenotype, and they're also associated with a greater response to immunotherapy, kind of already lending this idea that we can be targeting genetic alterations in this type of cancer. And another gene that's gaining traction in this field is PI3R1. And we can know this already just because PI3R1 mutations are very frequent in endometrial cancer. Using the cBioportal for Cancer Genomics database, we can take a look at the top tumor types that hold PI3R1 mutations. And so on the x-axis here, what we're looking at is different tumor types. And on the y-axis, we have the frequency of PI3R1 mutations within those tumor types. Now, just looking at this right away, we can see a whole bunch of different kind of cancer types, but these first 4 cancer types are really drawing our attention because they seem to be quite enriched. And if we kind of hone in on them, I'm not sure if you can really read it, but trust me in that they are all endometrial cancers. And we also, moving off of this, took a look at our own internal data with our patient cases and analyzing almost 4,500 patient cases that we've worked on. There are 346 of those cases that were endometrial cancer, and of those, 82 were PI3R1 mutated. And so taking this further, it reveals that we had roughly 24% of our endometrial cancer cases harboring a PI3R1 mutation. So it's really kind of cool to see that our own clinical cases are matching what's present in some of these cancer databases and even in the literature as well. So if we take a little closer at the genetic components of endometrial cancer and which genes are mutated, I'd like to represent that here with this figure from a meta-analysis of human endometrial cancer patients, where mutations in different genes on that y-axis were identified in differing stages of endometrial cancer. And we can kind of see, I want to bring your attention to the top here, these genes that are identified mostly in the earlier stage cancers, by and large, are members of the PI3 kinase signaling pathway. So we have PTEN, KRAS, PIK3CA, and then our gene of interest today, PI3R1. And not only is PI3R1 commonly mutated in endometrial cancers and very prevalent, but it also seems to have a link to prognosis as well. And we can see that here with this survival plot. Patients who were harboring a PI3R1 mutation, shown in blue, had a statistically significant lower overall survival rate compared with patients who had a PI3R wild type, shown in red here. So already we're like, okay, this is really prevalent, seems to have an effect on prognosis. Like, why is this gene so important? And we can get a better look at that by looking at the signaling pathways that it participates in within the cell. And I'll just cut to the chase here. It's such a big deal because PI3R1 interacts directly with the oncogene PIK3CA and the tumor suppressor PTEN. And PI3R1 achieves this kind of dual tumor suppressor role by interacting directly with these two proteins. And that's illustrated here with this cartoon. Looking at this, I did want to point out that P85 is the gene that PI3R1 encodes for, actually P85 alpha. And it's just simplified here in this figure to P85. If we start looking at the left side first, we can see that the PI3R1 protein is binding to a P110 subunit, and PI3R1 mainly binds to the P110 alpha subunit, which comprises the PIK3CA oncogene. So by PI3R1 binding to PIK3CA, PI3R1 is able to regulate the PIK3CA signaling and attenuate like extra oncogenic signals that it may be trying to push forward. And so it's able to restrict the oncogenic capacity of the PIK3CA protein. If we take a look at the right side, we can see that PI3R1 also binds directly to PTEN, one of the more famous tumor suppressors that we're probably familiar with. By binding to PTEN, PI3R1 is able to stabilize the PTEN protein and promote its tumor suppressive activity. And interestingly, in some in vitro lines, knockdown of PI3R1 also causes a decrease in the amount of protein or the amount of PTEN protein in the cell too. So PI3R1 is very critical to helping PTEN do its job. And to achieve this sort of homeostatic balance between suppressing the oncogenic side and stimulating the tumor suppressive side, PI3R1 achieves that through multiple different domain regions. So looking here at the gene map for PI3R1, we can kind of divide this into an N-terminal half and a C-terminal half to see what it's really doing and how it's achieving this dual interacting properties. So looking at the N-terminal side, you can see these two little domains in blue. The first domain is a Sark homology domain, abbreviated to SH3. And this darker blue domain is a breakpoint homology cluster region, shortened to just BH. These two domains together comprise the PTEN binding region of the protein. So this N-terminal side of the protein is associated largely with the direct PTEN binding that we were just looking at on the previous slide. If we take a look at the C-terminal half, we can see that there are more SH domains. We have an N-terminal SH2 domain, a C-terminal SH2 domain, and then the region lying between these two domains is called the inter-SH2 domain, or the ISH2 domain. Spanning across the N-terminal SH2 and the inter-SH2 domains, this is where the P110 or the PIK3CA binding region lies. And so we are already kind of seeing like, all right, half of this is P10, the other half is PIK3CA. We can kind of start imagining that if you have mutations kind of at any part of this gene, you might be messing up its ability to interact with PIK3CA and P10. And we can see that using the Mastermind Search Engine to look at literature of variants published in the PI3R1 gene. And so just to orient ourselves with what we're seeing here, this bottom portion is representative of the full length of the PI3R1 gene, and I've highlighted the PI3 and PI3CA binding regions just to kind of keep us on track of what I wanted to focus on with the PI3R1 today. And then the increasing heights of these bar graphs along the length of the gene are correlated with increased literature reported for variants at that location along the gene. And what we can really take home from this figure here is that there are a lot of papers published on PI3R1 variants, and they're not really necessarily clustered anywhere. They're kind of present along the whole length of the gene, indicating even further that this gene is very critical at multiple points. Taking a closer look at the PI3R1 variants themselves that are published within endometrial neoplasms specifically, we can see this kind of honed down a little bit. It's not as extreme as the previous figure, but we do still see that there are variants documented across the full length of the gene in endometrial cancer specifically. These variants that are coming up here at the bottom are some of the most published variants within endometrial cancers. And for the rest of the talk today, I wanted to hone in on 3 of these variants. This first variant up here is a truncation variant, and then we have a missense and a single amino acid deletion. Before I get into them each super in detail specifically, I wanted to draw your attention up to the top to this lollipop diagram of the PI3R1 gene. And this illustrates hotspot mutations along the length of the gene with increasing heights in the lollipop indicating increasing prevalence of that specific mutation. And the first one I want to call your attention to is the R348 truncation that we highlighted on the previous slide. This does correlate with that very tall lollipop right there, indicating that this is certainly a mutational hotspot. And if we kind of pay attention to where it's lying, it's a truncation downstream of the PI3 binding region, but upstream of the PI3CA region. So if we kind of start thinking like, what is this alteration going to do? We could already kind of start imagining that it would maintain PI3 binding, maybe lose the PI3CA binding and regulation. And now this is when I want to draw your attention to this little more like kind of chunky graph or table, but I wanted to call attention to the R348 variant here in this row. This full table shows not only what domain these different alterations in PI3R1 fall within, but it also illustrates its protein binding to either PI3CA or PI3, downstream signaling pathway activation, and then if it's was transforming in cell culture. And for this particular truncation, it kind of fits with what we would see, or what we would think would happen. Since we are truncating upstream of the PI3CA binding region, it's losing the ability to bind to P110, sorry. And then since it's retaining the P10 binding region, we still have binding to P10. Since it's losing the PI3CA binding region, we have increased phosphorylation of AKT and junk and ERK signaling, indicating that these downstream pathways that PI3CA would normally be held back from are just kind of going crazy without the functional PI3R1. These other 2 alterations aren't represented within this table, but we can sort of pull ourselves down here to look at these alterations down here that are within that same region of the latter part of the ISH2 domain, still within that PI3CA binding region. And these variants are still interestingly, the ones in the table are still interestingly shown to bind PI3CA, but they end up losing the regulation of the AKT pathway downstream and are transforming in cell culture. So what I really want to have us take away from this is that different types of alterations at different locations within PI3R1 have these kind of variable responses of PI3R1 activity within the cell. And it just kind of even lends more credence to the fact that these mutations should be probably studied very closely and individually, since they can have such different effects. So now we can take a look at these three that are enriched in endometrial cancer specifically. And I'd like to start with the missense of the lysine to a glutamate at position 567. Here, using the Mastermind search, I was able to get some really useful papers. And here I have representative information from 3 different papers. And we'll just kind of go through these a little bit one by one. At the far left, we can see a Western blot of the P85 alpha wild type, so the PI3R1 wild type in this second lane. And then we also have our variant of interest right now in this last lane. And if we kind of just move past this and track down to what I'm really trying to hit on here is these 2 indicators of AKT phosphorylation at 2 different residues. And from this figure, we can kind of appreciate already that there seems to be some increase in AKT phosphorylation between the wild type to the mutant, indicating that maybe this is losing perhaps some PI3CA regulation, which would fit with what we saw earlier. This is within that PI3CA binding region in the latter part of the ISH2 domain. So this is luckily fitting with what we would think would happen, and it's always nice in science when things happen that you think are gonna happen. So what a great day, right? Another interesting thing is this variant was studied in 2 different cell lines and was found to be activating and transforming in both. And then even further, I think what's really interesting on this slide is that this variant has been documented in an endometrial cancer patient that also had a brain metastasis. And interestingly, it was only identified in the endometrial cancer sample itself, which I can't speculate too much on what that means, but I think it's really cool that it came up just specifically within their endometrial tumor. The next variant I would like to talk about is the one just a little further downstream of this variant, the single amino acid deletion of a threonine at position 576, still within that latter part of the ISH2 domain. And from these 2 papers, I was able to find some interesting information that at this top left box, by and large, somatic mutations within that ISH2 domain of PI3R1 can largely be considered oncogenic driver events within the cell. And I think that this is particularly interesting given that endometrial cancer meta-analysis that I had up here earlier, where PI3R1 and other members of the PI3 kinase pathway seems to be overrepresented in the earlier stage cancers, meaning that perhaps mutations in this gene and the others in that pathway could be some of the culpable drivers in endometrial cancer. On top of that, we had some functional characterization within this paper that showed that this variant showed increased activity of PI3CA compared with other variants and the wild type PI3R1. And this also has been previously published as an oncogenic variant. And further in vitro data showed that this variant led to IL-3 independent growth. It was transforming in cell culture, and it also was increasing AKT phosphorylation. So I just think it's pretty crazy that deletion of a single threonine can be causing all of these problems downstream. And if we take a look at that final variant, that truncation at the R348, this variant's actually pretty interesting. It's the most common reported variant in PI3R1, and it accounts for 10% of all variants in PI3R1. Like, it's important, right? Like, it's a big deal, not only in endometrial cancers, but beyond. And so it does kind of fit that this variant is the one that pops up mostly in endometrial cancer queries. But from this paper, this sort of summarizes initially those results that I was discussing on the previous slide, that this variant leads to increased AKT phosphorylation, loss of regulation of the JNK and ERK pathways. But I think what's really interesting about this variant is, since it is so prevalent and it's gotten so much attention, it's been linked to the possibility of cancer patients harboring this alteration being sensitive to PI3 kinase inhibitors. We can now translate into the therapeutic part of this talk. I can move past my daily life of variant curation into how this could impact a patient and their signaling. So within this kind of simplified cartoon, we can see that this is representing the PI3 kinase AKT mTOR pathway. And I'll just call our attention to the PI3CA and the PTEN proteins that we've been talking about a lot today, these two, what's not illustrated between these two genes is PI3R1, but we can see how this all plays into downstream AKT and mTOR signaling just within this side of this figure. Now for what we do at Intermountain Health with our TheraMap testing is if patients have mutation, let's say in PI3CA or mTOR, these are two genes that have FDA-approved on-label therapy associated with them. Alpelisib and copanlisib are direct PI3CA inhibitors, and these three down here, sirolimus, everolimus, and temsirolimus, are direct mTOR inhibitors. And so these can be recommended to patients as on-label therapies, but with our TheraMap patients, we take a little bit of liberty and we try to provide our oncologists and treating physicians with as much information as possible that could help these patients. So when we encounter mutations within this pathway that are directly related to these two genes, we can then recommend these therapies listed here as off-label therapies, providing the oncologist with information that even though they don't have these specific PI3CA mutations or mTOR mutations that would get these assigned as on-label, they may have some benefit in attenuating portions of this pathway as a whole. And we're not the first ones to come up with this idea, right? That some of these other genes that don't have on-label therapy may be functioning to respond to some of these drugs. And it's pretty exciting that there are active clinical trials now that are recruiting patients with PI3R1 mutations to test some of these inhibitors that are kind of ancillary in its signaling pathway. The first one here we can see is a phase 2 study of that PI3CA inhibitor, copanlisib, that I was just mentioning. And you can see that this is recruiting patients with mutations within that pathway, the PI3CA, PI3R1, and PTEN as well. This second trial is a phase 1 trial that's testing ipatasertib, which is a pan-AKT inhibitor, so a little bit downstream of the PI3CA. Maybe if these drugs can't target PI3CA, maybe we can catch it further downstream. And this one is particularly interesting because this clinical trial is recruiting ovarian cancer patients, which I didn't mention today, but PI3R1 mutations are also enriched in ovarian cancer, not to as high a degree as endometrial cancers. But I think it's especially interesting because we've seen that trend of enrichment within our own ovarian cancer patients as well. So with that, I would like to thank you all for your attention today, and I'd like to acknowledge Intermountain Health and my department, Intermountain Precision Genomics, and all of the fantastic people on my team. In particular, I'd like to thank Anissa and Laura, who are here today, for helping me prepare these slides and for listening to me practice. And then I'd also like to thank our bioinformatics lead, Dustin Miller, who performed the analysis of our internal cases that we've worked on. And with that, thank you, and I'll take any questions.